we stopped here. I had to talk to somebody about something. So what traits do you have? This is just kind of something for fun. And I mentioned that usually there is a dominant or recessive trait, but it's not following Mendelian genetics most of the times because of the complications that we talk about and some of the exceptions. Okay, so just for fun, the first one, you can just pull your hair back and then see what kind of trait you have. Do you have a widow's peak, which is dominant, or do you have a straight hairline, which is recessive? And the second feature is here. You can probably guess freckles. Yeah, so you might not know, but having freckles is actually the dominant trait. And then the third trait is dimples. Yeah, so dimples are more complicated. Dimples are actually coded by multiple genes. And sometimes things can change too, right? Sometimes you had dimples when you were a child, and then as you grow, you lost the dimples. That's possible. Or you didn't have dimples when you were a child, and then when you're an adult, you started to have dimples. And sometimes you may have multiple dimples, right? This one is rolling the tongue. This is a very classic example when people talk about trait or the principles of inheritance. So if you can roll your tongue, that's dominant. And if you can't, that's recessive. I think I had a question in the lab test. This is very interesting. If you put your hand together, like in the picture, usually you will have a one hand or the one thumb over the thumb of the other hand. If you do it the other way, you're going to feel not very comfortable. It feels weird. And that's also determined by your genetic makeup. And then this one is similar, right-handed or left-handed. And you can guess that right-handed is really the dominant trait. Okay. All right, now Mendelian genetics. So these are going to be the exceptions that do not follow Mendelian genetics. And we're going to go over 3 main types of inheritance that do not really follow Mendelian genetics. And then we're going to have some questions, some practice questions for each type. And then in the study guide, I will give you some specific examples of genetic problems for you to solve. Okay? So the first one is called incomplete dominance. So incomplete dominance is the expression of 2 contrasting alleles, such that the individual displays an intermediate phenotype. So what does it mean? Basically, in this case, you do not have any dominant or recessive trait. So if you have 2 different alleles, you can still think of them as dominant or recessive, if that helps. If you have 2 different alleles, so in this case, if you have C prime, so C codes for red color and C prime codes for white color. If you have 2 different alleles, which you also say heterozygous, right? So instead of a red, which is often the dominant trait in the previous examples we talked about, so instead of a red, you will have a trait, a flower color that's intermediate in between red and white. So in this case, it's going to be pink. All right. So here, individual displays an intermediate phenotype. Because there's no dominant or recessive, all the alleles are equal in strength, and then they mix together. All right. So again, I have some flowers here. If you have CC, which is a red, let's see if I can erase that. There you go. And then you have C prime, that's white. If they cross, then it's 100% CC prime, right? And then we know CC prime is pink. How do you know? Because in the question, I will tell you that this follows incomplete dominance, right? Okay, so again, genotype, I put a table here to kind of help you figure out what the genotype and phenotype is. So we have, if CC, red, right? C prime, that's white. And CC prime is pink, right? Now, in this case, in incomplete dominance, there are a lot of hints there. So if I don't give you the genotype, if I just give you the phenotype, you should be able to figure out what the genotype is, because there's only one possibility. See, for each phenotype, there's only one possible genotype. So if I say, let me give you another example. So let's talk about hair. Okay. So one trait is curly hair. And then the other one is straight hair. And there is an intermediate trait, which is wavy hair. So if we use big W and little w for the different alleles, if I say a curly hair person and a straight hair person, oh, there's T missing there, they have children, what would be their child's genotype and phenotype? Okay, so curly hair, straight hair, cross, and what will be their genotype and phenotype? And I'm going to tell you that in this case, it follows incomplete dominance. It follows incomplete dominance. So you can figure out that curly hair, I'm going to tell you, W is curly and the big W curly, little w is straight. So curly hair, it has to be big C, big C. There's no other possibility, right? Can it be CC prime? No, because that's going to be the intermediate phenotype. It won't be curly anymore. All right, now straight hair has to be, oh, I'm using the wrong letters, right? This has to be big W, big W. For straight hair, it has to be little w, right? So can it be big W, big W? No, that's a curly hair. Can it be W prime? Nope, because W prime will be an intermediate trait, right? An intermediate phenotype. It wouldn't be curly hair, it wouldn't be straight hair, and instead it's going to be this wavy hair, all right? So just remember that for intermediate dominance, the difference is this heterozygous, not dominant anymore, right? Just heterozygous. If you have two different alleles, the individual is going to have an intermediate phenotype, something in between. All right, now the second one that we're going to talk about is codominance. So you know what co means, right? That means the two alleles that are present are both dominant alleles. So again, what does dominant mean? That means that allele is expressed, right? It's shown in the offspring. We are going to look at some examples. So I have some pictures here, the cat and the horse. So this is both about their fur colors. So their fur colors follow the pattern or the inheritance of codominance. So for the horse, you see there are two colors, right? So you see the gray color and the chestnut color, or I'm just going to say orange color. Okay, so these are actually coded by two alleles. We can say big G and little g. Okay, so let's say big G is for gray and G prime. Let's not use little g because this is not recessive. Okay? So little g for orange. So this horse, because it has both traits shown at the same time, so we know that this horse is going to be G prime, right? Because it has both colors. And it's actually the same thing for the cats. The cat has, again, black and orange, right? And it's also because the cat has two different alleles, and both alleles are expressed in the cat and the fur color. So that's why the cat has a patchy kind of colors. So that's the animals, and this also happens to flowers. I have a picture here just to kind of compare between incomplete dominance and codominance. I want to kind of help you figure out the difference. So remember, incomplete dominance, if you have a heterozygous genotype, then you are going to have an intermediate trait, right? So pink is going to be in between red and white. But for codominance, if you are heterozygous again, my dog is barking. If you're heterozygous, so instead of having an intermediate trait, something in between, you are going to have both traits shown at the same time, right? So you can see it's a patchy white and red. So you're showing both because both colors dominant. All right, so let's again practice filling up the table. For the color, you know what, just to kind of make it a little bit easier to understand and less confusing, we are going to use R for red. Okay. And then you can use maybe W for white, if that helps. Or you can use R prime for white, sorry. So that works too. Now, genotype, big R, right? So that's going to be red. So what about R prime? Right, that's going to be white. Oh, that's the key now. This is the most important one. What about you have R prime? What will be your color? Right, red and white. You have that patchy color, right? Another very useful example for codominance is actually human blood types. We did a little bit blood type in the blood typing virtual lab. So I will talk about more details when we do the lab test review. For here, just some really quick information. Type A, if you remember from the virtual lab, is really determined by the A antigen present on the surface of red blood cells. Right? A antigen. So again, what determines blood type? So for ABO system, if you are looking at a red blood cell, it really depends on what antigen you have on the red blood cell surface. Okay? So if you have A antigen, if this is A, then you're going to have type A. Okay? If you have B over here, B antigen, then you have a blood type B. If you have a type AB, that means you have both A antigen and B antigen present on the red blood cell surface. So if I have to draw, I'm going to use the triangle for B. Okay, so this is going to be B. So you can see you have both antigens present. So A and B antigen. What if you have blood type O? You don't have any antigen present on the surface. So you can see there's nothing there. So that be type O. Alright. When we look at blood types, there is another factor we need to consider, and that's the RH system. So that's the RH factor. So the RH is also a type of antigen. It's different than the ABO system, but it's also critical in determining people's blood and what kind of blood they can donate or receive. So the RH, like I said, is also an antigen on the surface of a red blood cell. And so I'm going to use the squares. So if you have RH antigen present, then you are positive. And that's where the positive negative sign comes from. It's determined by the RH system. If RH absent from your red blood cell surface, like this, no squares, that's going to be negative. Okay, so my blood type is B negative, so that means I have B antigen, I do not have RH antigen. Okay, so that's what it means. Alright, now let's go back to the table. So for the codominance, we are only concerned about the ABO system. Alright, so we're not going to consider RH factor in this example. So the antigens are coded by specific genes or alleles. Right? So for A antigen, you need allele A to code for the A antigen. So this is how we write it out. We use I to kind of present the allele, and then to indicate what kind of allele that is, because you have different forms, right? Different forms of alleles. We're gonna put letter A on the shoulder, right, as a superscript. Make sure you put it on the shoulder. And then the B antigen, that means you gotta have the B allele. For A and B, you guessed it, you gotta have both alleles present, right? You gotta have A and B. And what about type O? Now, the blood type actually follows both codominance and dominance recessive. Okay, so if you have O, you don't have any antigen, then you have this almost like a non or recessive allele. So we're just going to use lowercase I for this allele, this recessive allele. All right, now we know the allele that codes for a particular antigen. Now we're gonna figure out all the possible genotypes for each phenotype, right? What type of blood you have, that's a phenotype. And then we're gonna figure out what the genotypes are for each type. So if you have type A blood, remember you need 2 alleles, right? Because you have 1 allele you inherit from your mother, 1 allele you inherit from your father. So for type A, you can be homozygous dominant. So that means you have 2 A alleles. Is there another possible way where you can also have type A? Sure, you can be heterozygous, right? You have an A, the dominant A allele. You also have the recessive little I allele. So if you have either phenotype, you will have type A blood. So the same thing here. You can be homozygous dominant or heterozygous dominant. Very good. Now, which one are we talking about? Is the codominance? Is this one? Okay. If you have blood type AB, that means both A and B are present and they codominate. Okay, so you have both A antigen and a B antigen. And type O, right? Only one possibility. Yeah. Okay. So we have some questions in the study guide. So make sure you go through those questions, do your best, and then when we do the lecture test review, I will go through each answer or go through each question and give you the answers. Okay? Now the next one we're going to look at is sex-linked traits. So we'll probably mention that for the lab test too. This is actually not very hard. The only thing you need to remember, you have to remember, is that you gotta remember to put the alleles on the shoulder of the X chromosome, all right? Because the genes are carried by the X chromosome. So that's why we say the traits, the genes are linked to the sex chromosome, and mostly it's X-linked. And on the Y, make sure you don't put anything there, because a Y chromosome does not carry those genes. So you have to keep this a blank. So if you can remember to keep this part blank, then you can do a correct Punnett square, and you can figure out the genetic problem. All right, so make sure you keep a Y blank, because we are just gonna focus on the genes that are carried by X chromosome and not on Y. So you can't put any letters next to Y. You can't put anything next to Y. Alright, now let me explain why most of the genes are linked to X chromosome and not Y chromosome. So if you remember, chromosome 23 is the sex chromosome, right? If you have XX, you're a female, XY, that means you're a male. Now you probably noticed that the X chromosome is a lot longer than the Y chromosome, correct? Okay. Now we mentioned that sometimes the chromosome may carry over 1,000 genes. So if you think about it, there's going to be a lot of genes on the X chromosome that's not going to be on Y, because Y is really short. So that's why all the traits coded by these genes, which are not present on the Y chromosome, all these traits are going to be linked to X chromosome, because you can't find them on Y. All right? Okay, so hopefully that makes sense. And now let's look at some examples, and then we're gonna do the Punnett square for sex-linked traits. Right. Now, most of the times when we talk about sex-linked traits, we're interested in genetic disorders. So you can imagine that those genetic disorders are caused by mutations in the genes that are only carried on X chromosome. Right? Now, most of those genetic disorders are recessive genetic disorders. So that means we are kind of going back to the dominant recessive inheritance again. But you need to remember that those alleles are linked to X chromosome. Alright, first let's use hemophilia as an example. So people with hemophilia have trouble clotting their blood because they're missing some clotting factors. So if they are hurt, if they have an injury, then they will have a very hard time stopping the bleeding. So they will bleed a lot more than normal people, and if it's not stopped, in time, they could really bleed to death. So sometimes the situation can be really dangerous. So hemophilia is a recessive genetic disorder. So that means if you have two recessive alleles, so basically you have the recessive genotype. If you have two recessive alleles, then you are going to have the disease. Okay, so how can you get two recessive alleles? You get one from your father and one from your mother. Okay, so in this case, both parents are responsible for the child's disease. Okay, now let's use H. The big H for dominant, which is the good allele, right? The good allele. The little h, that's a recessive. And we know that's the bad allele, right? If you have two little h alleles, then you have the hemophilia, you have the disease. Now, this is linked to X, right? So we have to figure out how to write out the genotypes correctly. So if you are a healthy female, then you carry these two alleles, right, on the shoulders of X. You have one form of not showing the disease, one form of being healthy. That's when you have two dominant alleles, then you're definitely healthy. Okay. Now, what if you have one dominant allele and one recessive allele? Over here. Now, the dominant is gonna mask the recessive, right? So the female is still going to be healthy, but she is a carrier, right? Because she is carrying one bad gene, which could potentially be passed on to the offspring. So she's healthy, but she's a carrier. Now, if she has two recessive alleles, then she's definitely gonna be hemophilic. All right. Now, what about males? Can you write out the genotypes for males? What's the genotype for a healthy male? X. Is it big H or little h? Healthy, so you gotta have big H, right? All right. And then because it's male, so it's XY. Do you put anything in there? No. Leave it blank. Okay? Leave it blank. Do not write anything there. X, big H, Y. That's it. All right. So what's the other possible genotype? What else can you think of? X. Because the gene is only carried by X, right? So you just only need to look at what allele that the X chromosome can carry. So that's going to be little h, right? That's the second possibility. And again, keep a Y clean, nothing. So if a male has this genotype, is the male healthy or is he hemophilic? Hemophilic, right? Because it's a recessive genetic disorder, and you can have the disease if you only have one recessive allele, because in this case there's no dominant allele to kind of mask over this, right? So if you have one recessive allele and no dominant allele, you can also be sick. In this case, the male is hemophilic. Okay, let's practice one more because I'm really kind of worried that this is what's confusing to a lot of students. Okay, let's say colorblind. All right, let's just use big C for the dominant trait, which is the good. You're not colorblind. And little c, oh, you know what? Let's use B because it's easier to differentiate the lowercase and uppercase letter. Okay, so little b is recessive, and because color blindness is a recessive disorder, so we know that's the bad allele. Okay, so here, write out the genotype for color blind male and female. For carrier, male and female. And for healthy individuals, write out their genotypes for male and female. Okay, I have a little trick somewhere. So